Hello friends, today we are going to start with the next point, that is the types of protein. And now later we are going to study structure of proteins. So let us first start with types of protein. We have already studied the introduction part of protein, that is definition of protein, examples of protein, and the basic unit of protein. Now in the types, the first type of protein is globular protein. So why they are called globular? Because they are spherical in shape. Now why they are spherical in shape? The answer is the spherical shape is due to coiling of polypeptide. As you all know, proteins are polypeptides containing many peptide bonds. And when these polypeptide they get coiled around themselves, they form a spherical, a spear-like structure, and therefore they are called globule. Globule means spear. Therefore, proteins are called globular proteins. These proteins are generally soluble in water. Example is insulin, egg albumin, or serum albumin, or ligamellin. Ligamellin is a protein which is obtained in the pulse, that is डाळी म्हणतो आपण मराठीत. या सर्व डाळींमध्ये जे प्रोटीन्स आहेत इट इज कॉल्ड लिगॅमेलिन. Insulin is secreted by pancreas. Egg albumin found in the white of egg. So all these proteins are globular proteins. The second type of protein is fibrous protein. Fibrous means they have fiber-like structure. They have elongated rod-like structure. The shape of this protein is due to polypeptide remains parallel to each other. Two polypeptide chains, they remains parallel to each other, and they forms a ladder-like structure by using hydrogen bond, and therefore they are like a fiber. They never gets coiled up. Therefore they are not spherical in shape, but they are in a fiber shape. They are insoluble in water. Example is keratin. Keratin is a protein which is obtained in the hairs, nail, or wool, etc. Then second protein is myosin. Myosin is a protein of muscle. By using, you all see muscles is fiber-like structure. So keratin and myosins are fibrous protein, whereas egg albumin, insulin, they are globular proteins. So these are the main two types of proteins. First is globular protein, second is a fibrous protein. Now we are going to study the very important part, that is structure of proteins. Protein shows four types of structure: primary proteins, secondary proteins, tertiary proteins, and quaternary proteins, in which the secondary proteins are again classified into two groups: alpha helix proteins and beta plated proteins. We are going to study primary and secondary proteins now. So primary proteins. What are primary proteins? In primary protein, many alpha amino acids are linked by peptide bonds. When you see example, you can clearly understand. When many alpha amino acids are linked by peptide bond means amide linkage, that is CONH bond. They are represented by writing three letter symbols of amino acid separated by dashes. The N terminal is written at the left end. N terminal means the terminal containing nitrogen, NH group, and the C terminal, the terminal containing CO group, is written on the right hand side. This is the example. So many amino acids, they linked with each other containing peptide bonds. This C double bond or NH group is called peptide bond, and it is represented by ALA -GLY ACR. I have given table in the last lecture. Go through this table. Every structure is represented by a three-letter symbol, and this symbol is used to represent this primary protein molecules. Now the second type of protein is secondary structure of protein. It is a three-dimensional arrangement of protein chain. In this structure, hydrogen bonding is present between NH of one peptide linkage and CO of another peptide linkage. So, two types of secondary structures are found: alpha helix structure and beta plated structure. So, this is a fibrous type of protein containing hydrogen bonding. Now, let us first study what is alpha helix structure. When polypeptide twist into clockwise or right-handed spiral chain, it forms alpha helix structure. असं स्पायरल स्ट्रक्चर तयार होतं याच्यामध्ये. The CO group of first amino acid is hydrogen bonded with NH of fourth amino acid in chain. So first carbon, first CO, the CO group of first amino acid and the NH group of fourth amino acid, they forms hydrogen bond between them, and hydrogen bonds are parallel to helix structure, whereas R group remain outside the helix. You can see this diagram. The structure is spiral, and the spiral is right-handed side clockwise direction, and there is a hydrogen bond dot shown by a dotted line. You can see the CO group of first and NH of fourth amino acid, they form hydrogen bond. So there are many hydrogen bonding, and it shows a fiber type structure, coiled structure. This type of structure is called alpha helix, and in every coil there is a 3.6 molecules of amino acids. Therefore, it is shown by 3.6 residues. If you count CONH group in every loop, you can see there are 3.6, 3 CONH group and half NH group, or 3 CONH group and half CO group is present in every loop. So this type of structure is called alpha helix type of structure. So this is the first type of secondary type of proteins. Now second is beta plated structure, or it is also called beta plated sheet. It has a sheet like structure. Two or more polypeptide chain line up by side to form beta plated structure. दोन चेन रांगेत उभारल्यानंतर जसे दिसतील तशा टाईपचा हा स्ट्रक्चर आहे. The CO of one amino acid and NH of another amino acid, they lie in the plane. Hydrogen bonding between CO and NH forms between nearby amino acid. So this is the difference between alpha helix and beta plated. In alpha helix, the CO of first amino acid forms hydrogen bond with NH of fourth amino acid. But in beta plated, there is no such restriction. All nearby CO group and NH group, they form hydrogen bonding between themselves. And R groups are found above and below the plane of sheets. You can see in the example. You can see R groups are above the plane or below the plane, and all CO and NH, they are in the plane. And whenever NH group of first upper chain and the CO group of lower chain, they form hydrogen bonding between them. So there are 4 dotted lines are shown in the structure. This dotted line is a hydrogen bonding. So main difference between alpha helix and beta plated is alpha helix structure is a spiral or coiled structure, whereas beta plated is a sheet like structure. In alpha helix structure, the CO group of first forms hydrogen bond with NH of fourth amino acid. But in beta plated, all nearby CO and NH group they can form hydrogen bonding with themselves. So today we have seen two types of protein, that is globular proteins and uh, fibrous protein. Then we have seen structure of protein in which primary structure represented by two-dimensional figure, whereas secondary structure is represented by three-dimensional figure. In secondary structure, there are two types: alpha helix, which has a coiled shape, and beta helix, which has a ladder-like shape. In the next lecture, we will see the enzymes and the tertiary and quaternary proteins. It is reduced in your syllabus. Therefore, after primary and secondary, we are moving towards next point, that is enzymes. So today we will finish this lecture with this beta plated structure. Thank you.